Welcome back, students, to my YouTube channel. Today's topic of discussion is pure culture preservation. Why pure culture is so important for any microbiologist or any biotechnologist. Because pure culture is a sample of one type of microorganism that is grown in a controlled environment. This allows the scientists to study the organism in detail without any interference of other type of microorganisms. So let's start with the introduction part. When one's microbe has been isolated, they need to be grown in pure culture form. It becomes very necessary for the viability and purity of the microorganism. So we need to periodically transfer them into the fresh medium and allow them to grow in the new medium so that we can use them for the extended period of time. The transfer is always subject to aseptic condition to avoid any contamination of other microorganisms, to maintain pure culture for extended periods in a viable conditions without any genetic changes such as mutation, as well as avoiding its contamination is referred to as preservation. Is what are the application where we can apply the preservation of pure culture. So first and foremost application of preservation is in the field of academia. So every researcher, every faculty or every student of bachelor's and master's need to preserve their microbial culture for the longer period of time to perform certain type of experiments. Another application is in the field for the research purpose. Every research purpose or every research scholar or every researcher need to preserve their microorganism for the longer period. The third application is in the field of industry. Every fermentation industry, whether it is for antibiotic, whether it is for any fermented drink, wine, they needed a pure form of microorganism for fermentation process. The last and foremost important is the in the field of biotechnology. Every biotechnologist need a pure culture form of microorganism for their experiment. The principle of preservation. The principle of preservation is depend upon two techniques. One, when the microorganism is in the continuous metabolic state, and second one, when the microorganism is in the suspended metabolism state. The continuous metabolic state of preservation technique. In this technique, organism is preserved on nutrient medium by repeated subculturing. In this technique, any organism are stored by using general nutrient medium. Hence, repeated subculturing is required because of the depleting nutrients or dying of the microorganism. This technique consists of the five techniques. First is periodic transfer of fresh medium. Second is refrigeration. Third is trap culture technique. Fourth is storage in sterile soil. And fifth one is line suspension. Long-term preservation. The suspended metabolism type of preservation is also known as long-term preservation method, and continuous metabolic state preservation is also known as short-term preservation. In long-term state of preservation, the organism should be in the suspending metabolism state. We can preserve our microorganism in long-term method by drying them or storing them at the low temperature. This technique is consists of four other techniques. First is drying in vacuum. Second one is paraffin method, also known as oil method. Third one is lyophilization, also known as freeze drying. Fourth one is cryopreservation. The first technique is periodic transfer. It is the most basic technique which comes under short-term preservation, is used in all microbiology laboratory. In this technique, organisms grown in journal medium on plant incubated for certain period or at the certain temperature depending on the characteristic of selected microorganism, then they stored in a refrigerator. When the time of microorganism is completed, we need to subculture them or transfer them into the same medium or different medium. It depends upon your experiment as well. So what are the advantages of periodic transfer? It is a very simple method, so no special or expensive apparatus or instrument is required. It organism recovery by this method is very easy. And what are the some disadvantages of periodic transfer? Since it is a repeated subculturing method, it must be time consuming and difficulty to maintain pure culture for the longer term was very crucial for any experiment. So it can't be achieved by this method. Risk of contamination is very much high, and possibility of genetic changes due to development of certain variants or mutants or change in biochemical characteristic must be there. So we need to use or choose some technique by which these type of disadvantages can be avoided. So strains can be maintained periodically by preparing a fresh culture medium from the previous stock. The culture medium, the storage temperature and the time interval at which the transfer are made vary with the species and must be ascertained beforehand. The temperature and the type of medium chosen should be support a slow rather than the rapid rate of growth so that the time interval between transfer can as long as possible. Many of the more common heterotrophs remain viable for several weeks or months on a medium like Newton agar. Plants can be used for this periodic transfer technique and they can be incubated for 24 hours and can be stored in the refrigerator for a certain period of time. After that we have to subculture them on the transfer them periodically. Is refrigeration. It is also very basic technique. In this technique, we can store our agar plants or culture plate at the refrigerated temperature that is 0 to 4 degrees Celsius. By this method, we can preserve bacteria for 2 to 3 weeks and fungi for up to 1 month to 6 months. Molds can be stored on potato dextrose agar plate at the 4 degrees Celsius for 6 months to 1 year by using refrigeration method. The third and foremost important method in short-term preservation is glycerol stock. In this method, the colony grown in solid medium but scraped off using inoculating loop, and those colonies were suspended in the glycerol stock. The glycerol stock can be of any percentage, 30%, 50%, 80%. It's depend upon your microorganism or on your experiment. And after that, the glycerol stock vial should be stored at minus 28 degrees Celsius, and the culture should be preserved for at least 12 to 18 months. This is the procedure. How can we prepare a glycerol stock for the preservation? First, we will take, well, we have, if we have to prepare 30% of glycerol stock, we have to mix 30 ml of glycerol and 70 ml of water and autoclave that solution at 121 degrees Celsius for 15 minutes. In we have first then we have to prepare at least 1 to 2 ml or 500 microliter of glycerol stock in the microcentrifuge tube, which is also known as MCTs. Then we have to suspend microbial colony or any bacterial colony or fungal colony into that mixture, vortex that, and paraffin the screw the cap and paraffin it so that it can be sealed properly, and we have to keep that vial into the at the temperature of minus 20 degrees Celsius. For by this procedure we can preserve our bacterial or fungal culture for at least 2 years. The basic method in short-term preservation is step culture. These cultures and preserve them at the room temperature, and these culture are used for non-fastidious organisms which do not have any spatial requirement, such as Staphylococci or Enterobacterus species. Prepare a tube with deep butt of carbohydrate-free agar such as tryptic soy agar. Swab the organism into the agar by using sterile inoculating needle. Incubate that tube at 35 degrees Celsius. Close the tube and we can store that tube at the room temperature or in the freezer as well, and the culture will be preserved for at least 1 year. Step culture in cystine trypticase agar method is recommended for Neisseria and Streptococci species. Another method are preservation of microorganism in sterile soil and saline or NaCl suspension. How we can preserve microorganism in sterile soil? In this method, we have to take a soil and autoclave it for 3 days so that any microflora or any microorganism in the soil can be killed completely. This method is widely used for the spore forming bacteria and fungi. In this method, organism will remain in dormant stage in a sterile soil. Sterile should be sterilized, then spore suspension added to it aseptically. This mixture will dried at room temperature and stored in refrigerator, and by this method the viability of microorganism found to be around 70 to 80 years. And second method is preservation in saline suspension. Sodium chloride in high concentration is frequently known to be inhibitor for the bacterial or fungal growth. Bacteria are suspended in 1% salt solution, then the tube stored at room temperature, or whenever needed the transfer is made on the agar saline. Normal saline used to provide proper osmotic pressure to the organism. Otherwise, high salt concentration is inhibitory for organism. Organism kept in screw cap bottle in normal saline, stored at room temperature whenever or wherever required, transfer made on agar saline and incubated. Till now, we have only talked about how we can preserve any aerobic microorganism, but we have to preserve anaerobic microorganism as well. So for that purpose, cooked meat medium should be used for the preservation of anaerobic microorganism. Inoculating tube of cooked meat medium with the isolate, incubate them overnight at 35 degrees Celsius, close the tube with screw cap or cork and store at the room temperature. Transfer should be made every 2 months. Now I will tell you about the long-term preservation method. The first method in long-term preservation is drying in vacuum. This is the basic method. In this technique, organism dried over chemical instead of air dry. The cell passed over calcium chloride in a vacuum, then stored in a refrigerator. By this, organism must survive for the longer period of time. As you can see in this picture, this is the type of apparatus which is used for drying in vacuum. This is for use for the cell drying in a vacuum chamber. This is the vacuum chamber. This is the vacuum pump used for creation of vacuum. This is air dryer through which air is passes. This is the pressure transducer. By using this, we can apply pressure so that there won't be any air in this chamber. Third, in long-term preservation is overlaying culture with mineral oil or paraffin method. In this, organisms are grown on agar slant, then they are covered with this sterile mineral oil to the depth of 1 centimeter above the tip of the surface. This method is very simple. One can remove some organism in a septic condition with the help of sterile lube, and by this we can preserve our microorganism up to 15 to 20 years. Agar slants are inoculated and incubated until the growth appears. Many bacterial species can be preserved by covering the growth on agar slant with this sterile mineral oil. They are then covered with this sterile mineral oil up to the depth of 1 centimeter above the tip of agar slant. Oil must cover the slant completely. Oil, what will oil do? Oil reduces the loss of water and also slows down the exchanges of gases within the organism and surrounding. This type of method is very simple and most economical method for preserving any bacteria and fungi where they remain for the viable uh, for the several years and at room temperature. The layer of paraffin also prevents the dehydration of media by ensuring an aerobic condition. The microorganisms remain in the dormant stage so that microorganism can be preserved for the longer time. The advantage of this method is that we can remove some growth under the oil with the transfer needle and inoculate them on the fresh medium and still preserve the original culture. The simplicity of the methods makes it attractive, but changes in the characteristic of strain can also occur here. Now come to the procedure part, how we can prepare the tube or the microorganism for the paraffin method. Prepare a tube containing hard infusion agar with the short slant. For the fastidious organism, add fresh native or heated blood because fastidious organism needed some extra growth factors such as blood or any serum. Sterile mineral oil or petroleum in a hot air oven at 170 degrees Celsius for at least 1 hour. Grow a pure culture on agar slant. When the growth is seen at soil mineral oil about 1 centimeter above the tip of the slant, then subculture it. When needed, scrape the growth under the oil using sterile inoculating needle and store them at the temperature and transfer after 6 to 12 months. Method is lyophilization or freeze drying method. Lyophilization is vacuum sublimation technique, cell grown in nutrient media, and then this culture distributed in small vial. These vials then immerse in a mixture of dry ice and alcohol at 78 degrees Celsius. Then these vials immediately connected to a high vacuum line, and when they are completely dried, each vial sealed under vacuum. This is the most effective and widely used procedure. The principle of freeze drying is that freeze drying is a process where water and other solvent are removed from the frozen product via the process known as sublimation. Sublimation occurs when frozen liquid goes directly into the gaseous state without entering into the liquid phase. This is recommended using a slow rate of cooling, as this will result in the formation of vertical ice crystal structures, thus allowing for more efficient water sublimation from the frozen product. Freeze-dried products also are hygroscopic in nature. Hygroscopic means they do not require water. If water come in their presence, that water can harmful for the preservation process. So we need to protect the hygroscopic products from the moisturizing condition. Under these conditions, the microbial cells are dehydrated and their metabolic activity are stopped. As a result, the microbe go into the dormant state and retain the viability for years. Lyophilized or freeze-dried pure culture and then sealed and stored in the dark at 4 degrees Celsius in refrigerator. Freeze drying method is the most frequently used technique by culture collection centers. Many species of bacteria preserved by this method have remained viable and unchanged in their characteristics for more than 30 years. Freeze drying process results in a stable and readily dehydrated product. This process consists of three steps. First is pre-freezing state. Pre-freezing the product in a laboratory freezer to form frozen structure. This should be done at minus 20 degrees Celsius. Then second step is primary drying to remove most water. And third step is secondary drying to remove bound water, as you can see in this picture. First is the freezing state, second is primary drying stage, third one is secondary drying stage. In the first stage, the atmospheric pressure is applied. This should be done at minus 40 or at this minus 20 degrees Celsius temperature. In the first step there is a formation of crystals. In second step there is a sublimation of ice crystal, and in third step there is a decrystallization of the surface water. This is very important, how we can recover bacteria when the organism is in the lifeless state. For this, we have to remove the frozen culture from the freezer, then place them on a dry ice or into the alcohol or dry ice bath. Transfer to the laboratory in a safety cabinet, and the cabinet should be clean. Scrape the topmost portion of the culture using style low. Transfer that culture to the growth medium without contamination. Reclose the vial before the content completely thaw and return the vial into the freezer carefully. And we, after that we can incubate that tube at the for the 18 to 24 hours at 35 degrees Celsius and perform at least one subculture before using the isolate to inoculate test. What are the advantages of lyophilization? Lyophilization, there only minimal storage space is required. Hundred of lifeless culture can be stored in a small area. Small vial can be sent conveniently through the mail to the other microbiology laboratory when packed in a special sealed milk container. Lifeless culture can be revived by opening the vial, adding the liquid medium, and transferring the rehydrated culture to a suitable growth medium. These are the advantages of lyophilization. Last procedure is cryopreservation method. The cryopreservation, that is freezing in a liquid nitrogen at the temperature of minus 196 degrees Celsius, or in the gas phase above the liquid nitrogen at the 150 degrees Celsius. This helps the survival of pure culture for the long storage time. In this method, the microorganisms of culture are rapidly frozen in a liquid nitrogen at minus 196 degrees Celsius in the presence of stabilizing agents such as glycerol or DMSO. These stabilizing agents prevent the cell from the damage due to formation of ice crystals and promote the cell for the survival. This liquid nitrogen method has been successfully used with many species that cannot be preserved by lyophilization, and most of these species can remain viable under the for at least 10 to 30 years without undergoing any change in their characteristics. However, this method is very expensive due to the liquid nitrogen use. This is the protocol of cryopreservation. This is the sample. First, we have to filter our sample using the syringe filter. Then, if the sample is liquid or if the sample is concentrated, we have to mix our sample with the cryoprotectant such as glycerol or DMSO. These cryoprotectant will prevent the cell from getting damaged from the ice crystals. Then the final volume of the liquid inoculum, whether it is in the non-concentrated one, whether it is in the concentrated form, the final volume should be 5% only. The After that we have to equilibrate that tube at 4 degrees Celsius for 15 minutes. Then we have to flash freezing the sample using liquid nitrogen, and then we can store that tube at minus 80 degrees Celsius. If we have to revive that sample, just take it out that vial from the minus 80, just throw it, and just after using it, put it back into the minus 80 degrees Celsius, otherwise your cell will get damaged. If you have liked my videos, please do subscribe my channel for more such videos, and do not forget to like my video. Thank you.